Cellular respiration is the process by which your body converts biochemical energy from nutrients in the food you consume into energy that's usable by the body. It's the broad term that describes a set of metabolic reactions and processes that occur in the body that allow us to utilize food as an energy source. Glycolysis is the pathway that converts sugar into energy or glucose C6H12O6 into pyruvate, generating ATP during the conversion. An important term to know is catabolism. Catabolism is the breaking down of larger molecules into smaller ones. Anabolism is the building of larger molecules from smaller ones. The truth is, in the name glyco means glucose and lysis means to unbind. Glycolysis literally means breaking down glucose. Glycolysis is catabolic. It breaks down glucose, a 6-carbon sugar, into pyruvate, a 3-carbon sugar. These are the glycolysis steps. Step number 1. Phosphorylation of glucose. In the first step of glycolysis, the glucose is initiated or primed for the subsequent steps by phosphorylation at the C6 carbon. The process involves the transfer of phosphate from the ATP to glucose, forming glucose 6-phosphate in the presence of the enzymes hexokinase and glucokinase. This step is also accompanied by considerable loss of energy as heat. Step number 2. Isomerization of glucose 6-phosphate. Glucose 6-phosphate is reversibly isomerized to fructose 6-phosphate by the enzyme phosphohexoimerase. This reaction involves a shift of the carbonyl oxygen from carbon 1 to carbon 2, thus converting an aldose into a ketose. Step number 3. Phosphorylation of fructose 6-phosphate. This step is the second priming step of glycolysis, where fructose 6-phosphate is converted into fructose 1 and 6-biphosphate in the presence of the enzyme phosphofructokinase. Like in step number 1, the phosphate is transferred from ATP while some amount of energy is lost in the form of heat as well. Step number 4. Cleavage of fructose 1 and 6-diphosphate. These steps involve the unique cleavage of the CC bond in the fructose 1 and 6-biphosphate. The enzyme fructose diphosphate aldolase catalyzes the cleavage of fructose 1 and 6-biphosphate between C3 and C4, resulting in two different triose phosphates, an aldose and an aketose. The remaining steps in glycolysis involve 3 carbon units rather than 6 carbon units. Number 5. Isomerization of dehydroxyase phosphate. Glyceraldehyde 3-phosphate can be readily degraded in the subsequent steps of glycolysis, but dehydroxyase phosphate cannot be. Thus, it is isomerized in glyceraldehyde 3-phosphate instead. In this step, dehydroxyase phosphate is isomerized into glyceraldehyde 3-phosphate in the presence of the enzyme triose phosphates isomerase. This reaction completes the first phase of glycolysis. Step number 6. Oxidative phosphorylation of glyceraldehyde 3-phosphate. Step 6 is one of the 3 energy-conserving or forming steps of glycolysis. The glyceraldehyde 3-phosphate is converted into 1,3-biphosphoglycerate by the enzyme glyceraldehyde 3-phosphate dehydrogenase. In this process, NAD+ is reduced to coenzyme NADH by the H from glyceraldehyde 3-phosphates, since 2 moles of glyceraldehyde 3-phosphate are formed from 1 mole of glucose. 2 NADH are generated in this step. Step number 7. The transfer of phosphate from 1,3-diphosphoglycerate to ADP. This step is the ATP generating step of glycolysis. It involves the transfer of phosphate group from the 1,3-biphosphoglycerate to ADP by the enzyme phosphoglycerate kinase, thus producing ATP and 3-phosphoglycerate. Since 2 moles of 1 and 3-biphosphoglycerate are formed from 1 mole of glucose, 2 ATPs are generated in this step. Step number 8. Isomerization of 3-phosphoglycerate. The 3-phosphoglycerate is converted into 2-phosphoglycerate due to the shift of phosphoryl from group carbon 3 to carbon 2 by the enzyme phosphoglycerate mutase. This is a reversible isomerization reaction. Step number 9, dehydration 2 of phosphoglycerate. In this step, the 2-phosphoglycerate is dehydrated by the action of enolase 2-phosphoenol pyruvate. This is also an irreversible reaction where 2 moles of water are lost. Step number 10, transfer of phosphate from phosphoenol pyruvate. This is the second energy generating step of glycolysis. Phosphoenol pyruvate is converted into an enol form of pyruvate by the enzyme pyruvate kinase. The enol pyruvate, however, rearranges rapidly and non-enzymatically to yield the keto form of pyruvate. The keto from predominates at pH 7.0. The enzyme catalyzes the transfer of phosphoryl group from phosphoenol pyruvate to ADP, thus forming ATP. The overall process of glycolysis results in the following events. Number 1, glucose is oxidized into pyruvate. Number 2, NAD plus is reduced to NADH. Number 3, ATP is phosphorylated into ATP.